In this video, we're going to be demonstrating an enzyme-linked immunosorbent assay, or an ELISA. This one in particular is the TSH assay. So we've let the reagents come to room temperature. We've reconstituted our standards. We have our QC material as well. And now we are going to pipette our samples. We have our microtiter plate, and these are special wells that are coated with antibodies to TSH. So when you're pipetting, make sure you don't scratch the tip of your pipette on the bottom of the well, because it will scratch off the antibody. So first thing we're going to do is pipette our standards and our QCs, and then finally our little sample. And when I'm pipetting, I am resting the tip along the sidewall and changing tips between each sample, even though I'm going low to high, because we don't want any cross-contamination. I'm pipetting 100 microliters. And when you do the blowout, you might get bubbles, but that is okay because they will pop, hopefully. And once you're done pipetting, you would record where everything is located on your template, which should be in your assignment. Your assignment for this lab, so that you know where everything is. So when you do your data analysis, you don't have any patient misidentifications. All right, next we are going to pipette 100 microliters in the conjugate. It's this nice red color for this lab, into each assay, into each well. And because I want to prevent cross-contamination, I will change tips between each well. When you're working with very tiny concentrations of hormones, any contamination can be significant. Alright, done that, and now we're going to swirl it for 30 seconds on the bench top so it doesn't splash. All right, now we're going to cover it with a plastic cover or aluminum foil and let it incubate at room temperature for 60 minutes. So we'll set a timer for that. And we will meet you back after this has incubated. So we just finished our 60-minute incubation of our enzyme conjugate with our sample. So now what we're going to do is we are going to discard the reagent that's in here by carefully flicking it into the biohazardous waste and not splashing ourselves. All right. All right, so now it's empty. You don't want to let this sit and dry out, so we're going to move on to our next step, which is adding 200 microliters of distilled water to each well. And this is our first rinse step. So the goal of this is to get rid of any unbound enzyme conjugate. And I am not actually touching my tip to the inside of this well so that I don't have to change tips. If you don't trust your skills, then you can touch the tip and change the tips between each one. Now I'm going to flick it again. And I have a little aluminum foil here. I'm going to set down so I don't get my bench top super wet. I'm going to take this pad of paper towels and just tap, tap, tap out the excess. Discard the top one and fill it up again. So that was rinse 1, and we're going to do this 5 times. Alright, so now that we're done washing, we'll move on to the next step. And insufficient washing can cause inaccurate results. This is probably the most important step in your ELISA assays. So make sure you're doing it carefully and you're not skipping any wash steps. Alright, so we've washed. Now we are going to add our TMB reagent. And this is what will react with the enzyme linked to our antibodies that were in the conjugate reagent, and will give us a nice color change. So we are going to add 100 microliters. To each well, and use a new tip between each sample to prevent cross-contamination. And since this is clear, it's helpful that I have this on a sheet of paper so that I can see where I've already put stuff. I'm also not opposed to talking to myself and naming out what letters I've already done, because the well plate is labeled A through H. All right, so now that we've added that, we're going to do the circular mixing again for 30 seconds. Cover it again. And we're going to incubate this for 20 minutes. So this is now incubated for 20 minutes. I covered it with foil, so it likes to be protected from light. And we can see that it's changed to a nice little pretty blue color in some of our wells. So now we're going to add this stop solution. This is an acidic stop solution. What it does is it denatures the enzyme that is causing that blue color. And since it's an acid, it's going to change the pH, and that will cause this to turn yellow. So we're going to add 100 microliters to each well, and changing tips between them to prevent cross-contamination. And our next step is you can see that we've got this nice little yellow color. We're going to mix it on the bench top for 30 seconds. And if you didn't get any color change, that would indicate that there's been an error and that you'd need to repeat this assay with new strips. All right, so now we're going to take this over to the plate reader and read the absorbance at 450 nanometers.